All right, so first up today we have Justin Stilwell. He received his DVM and certificate in aquatic animal health at the University of Florida College of Veterinary Medicine in 2016. He then completed an aquatic-focused anatomic pathology residency at the University of Georgia in 2019 and earned his board certification in anatomic pathology in September of 2019. Justin currently serves as a diagnostic pathologist in the Department of Pathology at UGA and is currently finishing his PhD in veterinary pathology under the guidance of Dr. Alvin Camus of UGA and Dr. Matt Griffin of Mississippi State University. His doctoral work focuses on the pathology, pathogenesis, and metagenomic characterization of myxoid infections in fish, with an emphasis on proliferative gill disease in catfish aquaculture. So take it away, Justin.
All right. Thank you very much, Nick, for the introduction. I'd also like to thank Dr. Phelps, Dr. Griffin, and the American Fishery Society Fish Health Section for putting on this great seminar series. It's been a nice way to end the week each week during COVID. Today, I'll be presenting on one of the main projects of my dissertation research, examining how fish host susceptibility influences the myxozoan community composition of PGD outbreaks in catfish aquaculture. So we've heard several talks this series on myxozoans from Benjamin Amicus last week and Ethan Woodyard a few weeks prior. They gave great talks and really nice background overviews on myxozoan parasitism as part of their introductions. So briefly, myxozoans are morphologically diverse multicellular parasites that are distantly related to jellyfish and corals. They primarily infect fish, but reports also exist in amphibians, reptiles, waterfowl, and small mammals. And while most of these infections are incidental in nature, several are associated with economically important diseases, of which I'll be discussing proliferative gill disease. So proliferative gill disease affects catfish aquaculture, which represents the largest sector of food fish aquaculture in the United States, generating over $300 million in annual revenue. The industry grows channel catfish and a hybrid catfish, the latter of which have improved growth performance and resistance to several common channel catfish diseases. Production is limited by infectious diseases though, and myxozoan parasitism is an important one. So proliferative gill disease, which has been around since the 1970s, is among the most important infectious diseases of catfish. It causes high morbidity and mortality of all age classes, but particularly affects fingerling-sized catfish. The disease has seasonal outbreaks, most severely in the spring, and is caused by the myxozoan parasite Haenegaia ictaluri, which has an indirect life cycle involving the eligocete worm Diro digitata and the channel catfish Ictalurus punctatus. So I wouldn't be able to do this research if I weren't standing on the shoulders of giants, and many people since the 70s and 80s have done a tremendous work on studying this disease. To recap some of the highlights more recently, Dr. Linda Pope, picture in the top right, did the initial characterization of the parasite and did much of the early epidemiology work on the disease. Dr. Matt Griffin in the middle, my major co-advisor for my doctoral work, developed a qPCR assay for identifying the parasite within gill samples and pond water environments. His work also revealed a variation in the host species susceptibility to PGD between the channel catfish, the blue catfish, and their hybrid cross. And my immediate predecessor, Dr. Graham Rosser, showed that hybrid catfish have decreased rates of infection and arrested development of the parasite in the hybrid catfish. But despite these findings, PGD is still seen in hybrid catfish in the industry. Additionally, several other gill-infecting Haenegaia species of unknown clinical significance have been described over the past few decades in the catfish. So these findings put new focus on how the fish host species susceptibility shapes the myxozoan communities of catfish aquaculture ponds and PGD cases. To examine this question, our null hypothesis assumed that only assume that fish host species has no influence over the overall mixozoan community composition or diversity, while our alternative posits that fish host monoculture does influence mixozoan communities. Additionally, we wanted to investigate whether other Hennegaia species are potentially involved in PGD outbreaks. We know the disease is caused by Hennegaia ictalori, so our null hypothesis assumes that only it is involved in disease outbreaks. Our alternative hypotheses suggest that multiple species are involved, with Hennegaia ictalori always present, and either the most prevalent or not most prevalent mixozoan in the samples. To answer these questions, we conducted 2 experiments. The first involved collecting gill tissues for 3 years from diagnostic case submissions with gross and microscopic evidence of PGD. We also confirmed the fish host species by previously published PCR protocol and removed any cases that had mixed catfish species from the analysis. The second experiment involved using DNA from a pond water study previously conducted by Dr. Griffin and colleagues. What they did was they dedicated 18 ponds to either channel or hybrid catfish monoculture for 3 continuous production cycles. They collected water from each pond every 2 weeks over the spring PGD season, extracted the DNA, and used qPCR to compare the levels of Hennegaia ictalori abundance to severity of lesions seen in sentinel fish. And so we used the pond water DNA from that study for metagenomic analysis of a controlled pond monoculture environment. So extracted DNA from 342 gill samples and 342 pond water samples were submitted for targeted amplicon sequencing of the mixozoan 18S ribosomal RNA gene. Paired reads generated by Illumina MiSeq were merged, trimmed, filtered by quality and sequence length, and binned into operational taxonomic units using the usearch program. OTU sequences were then identified using a BLASTENG search of GenBank, aligned using the MAFT program, and then submitted for phylogenetic analysis using the MegaX software program. OTU counts, proportions, and relative abundances were used to compare mixozoan community composition, diversity, species evenness, and individual taxa prevalence between channel and hybrid catfish species and year-over-year trends within catfish species. All analyses were performed in RStudio using Permanova, Dirichlet multinomial modeling, Shannon diversity index, 2-sample independent t-tests, one-way ANOVA with p-value correction using the Benjamini-Hochberg methods for multiple tests. These metagenomic studies can have certain limitations, with primer choice being particularly important in designing these targeted approaches. And special care was taken to make sure that the primers did not overly select for one mixozoan versus another. Other things that we couldn't control because they came from diagnostic case submissions, we could not control for the age or size of fish, the time of season in which the fish presented, the time point in the disease progression, the possibility of mixed catfish species in the ponds, as well as potential undersampling due to pond-side diagnosis by farmers. The pond water experiment, on the other hand, limited or controlled a lot of these factors, but uneven actinospore distribution in the ponds remains a consideration. The water was also not filtered due to the abundance of plankton and the fine mesh size needed to filter out actinospores. Thus, the reads that we generated with the pond water study represent actinospore equivalents and not necessarily viable actinospores. So with that, I will start with presenting the data from the industry PGD gill samples. So the mixozoan communities significantly differed between channel and hybrid catfish PGD cases in all 3 years examined. These graphs are non-metric multidimensional scaling plots, which are ordination plots that help us visualize the beta diversity and community composition of each sample, which are the purple numbers in red regions for channel catfish samples, the green numbers in yellow regions for hybrid catfish samples, and regions of overlapping similarity in orange. And while there is some overlap in each year, gill samples exhibit considerable variability in their mixozoan community composition, often with no similarity to the opposite fish host. Looking at the channel and hybrid catfish PGD cases, this bar chart shows the average relative abundance of each mixozoan species on the y-axis, the different the catfish species, year and number of cases along the x-axis, and then the different mixozoans in the different colors. Looking at individual taxa prevalence, channel catfish had greater relative abundance of Hennegeia ictalori in 2017 and 2019, and of unclassified mixozoan taxa in 2018 on average compared to hybrid catfish PGD cases. There were no significant differences in channel diversity or species evenness between the channel or hybrid catfish cases, and no significant year-to-year trends within species. If we break these data down by individual cases, we have two major findings, with the first being that Hennegeia ictalori was always present in each case, and it was never the only mixozoan detected. The second finding is that while Hennegeia ictalori is present in all the cases, it is not necessarily the most prevalent mixozoan in nearly half of both channel and hybrid cases, as indicated by the blue boxes. And so these findings support those seen from the NMDS plots, where PGD cases exhibited considerable variability in the mixozoan community structure. So the findings in the industry gill samples were supported by those seen in the controlled pond water experiment. So I'm not going to present NMDS plots for this data, but for similar to the gill tissues though, the mixozoan communities in the pond water samples differed significantly between the channel and hybrid catfish ponds in all 3 years examined. And so these graphs are similar to the previous bar charts, with the relative abundance of the mixozoans on the y-axis, the sampling dates along the x-axis, which are divided into the 3-year periods. And then the different color lines representing the different mixozoans. And we can see the different fluctuation in abundance over time. So in year 1, all ponds experienced large peaks in Hennegeia exilis relative abundance, followed by more stabilized patterns in years 2 and 3. For channel catfish ponds, Hennegeia ictalori showed distinct peaks with significantly greater relative abundance compared to the hybrid ponds in years 2 and 3. For the hybrid catfish ponds, Hennegeia ictalori did not exceed 20% average relative abundance at any point during the study. And hybrid ponds also had distinct peaks of Hennegeia exilis relative abundance, which were significantly greater compared to the hybrid catfish, or compared to the channel catfish ponds in years 1 and 3. These findings are consistent with previous work with Dr. Griffin's previous study, and support the results seen in the gill tissues, suggesting hybrid catfish monoculture suppresses Hennegeia ictalori relative abundance in ponds and PGD cases. Channel diversity and species evenness were significantly greater in channel catfish ponds compared to hybrid catfish ponds in all 3 years examined, except for species evenness in year 2, which suggests that monoculture of hybrid catfish may lower the mixozoan diversity of the pond system. Lastly, these are phylogenetic trees generated from all of the known and unknown mixozoan sequences detected in the metagenomic samples, which illustrate the potential considerable mixozoan diversity that remains to be described in PGD cases and catfish ponds. Furthermore, nearly all of the catfish pond water OTUs were present in the gill samples, providing some validation for the existence of these taxa. So the results of this work substantially alter our understanding of proliferative gill disease of cultured catfish, demonstrating how fish host species provide selection pressures for mixozoan community composition and diversity in the disease process and the pond environment. Additionally, we showed that PGD cases may represent mixed species infections, with Hennegai ictalori always present, but not necessarily the most prevalent myxozoan. The abundance of novel unclassified myxozoan sequences in the ponds and gill tissues suggests substantial myxozoan diversity remains to be identified in the industry. Using this knowledge of how fish host species influences the myxozoan community, future work will focus on the development of crop rotation strategies, whereby rotating between channel and hybrid catfish may prevent any single myxozoan from dominating a system, and thus potentially preventing or mitigating disease outbreaks. And now that we've shown that PGD cases potentially represent mixed species infections, the final project of my dissertation will involve or investigate how myxozoans, aside from Hennegai ictalori, contribute to disease pathology by developing myxozoan species-specific in situ hybridization techniques and applying them to both challenge studies and industry PGD cases. But beyond catfish aquaculture, is there a bigger picture for this work? In sampling the pond environment, we were able to detect myxozoan sequences from other fish that occur incidentally in the ponds. These sequences had substantially lower relative abundance compared to the catfish myxozoans and did not significantly alter myxozoan communities in the ponds. But this does suggest that the myxozoan communities of these ponds reflect the ponds or any other water body really, likely reflect the hosts that are present where the life cycle can be completed for the parasite. It also suggests that host biomass might influence myxozoan relative abundance in the environment. And while there is some work to support this, it requires further investigation. And so finally, I'd like to acknowledge all of our collaborators for their contributions to this work, along with Dr. Michael Colvin, whom we consulted on the statistics, my wife Natalie in the UGA Pathology Department for their support over the last 4 years, and the various funding agencies that supported this work, including Mississippi State, the USDA, and the National Center for Veterinary Parasitology. These are my references. And with the last little bit of my time, I just want to give a quick shout out. My time as a graduate student in Dr. Camus's lab is nearing its end, and my position here is now being advertised, including in the Fish Health Section's newsletter that went out yesterday. So if you know any veterinarians or fourth-year veterinary students interested in pursuing aquatics-focused anatomic pathology, please spread the word. The program is terrific. Al is an amazing mentor to work for and a wealth of knowledge to learn from, and I can't recommend it enough, sorry. Yeah, feel free to contact me with any questions and I will take questions on my talk.
Awesome presentation, Justin. And I'm not sure if that picture's from a regular Friday afternoon in the Camus lab or Halloween, but looks like you guys have a good time down there.
Halloween, yep.
So you go ahead and unmute yourself if you got a question for him, or you can put it in the chat box and I can read it.
Hey, Justin, nice job on the presentation. This is Jan.
Hey, how's it going?
Good. I'm just curious with the different sequences that you're seeing out there. Some of these are uncharacterized species as well. Do you have any indication, do you think that these are all different myxozoan species or are some subspecies of Exilus or ictaluri? So what kind of like sequence divergence are you seeing between these?
So basically what we called, so we targeted a hypervariable region of the 18S. I think it's variable region 3. And basically we used the cutoff of 95% over that region. So we had done a sequence identity matrix beforehand to see of what we knew, what was the most closely related any two myxozoans were out of the described species. And the highest number we came up with was 92%. So we used 95 as sort of a safe cutoff. So all of these were under 95% similar to each other. I'd say most were in the mid to upper 80s, a few in the low 90s. So I think a lot of these are unique. That being said, maybe some are potentially similar to those already described. I know Ethan's post-Exilus is very prevalent in these samples, but we don't have molecular sequence on it until now. So that basically, I made those trees before we knew that really. So I think some of these are definitely unique species. So I think there is quite a bit of diversity out there that remains untapped or undescribed.
Yeah, really cool to see that sequence divergence and different species out there. Thanks.
Yep.
So with the catfish ponds, a lot of aquaculture is going towards all-in, all-out aquaculture just to prevent like pathogen replication and avoiding pathogens. Be extremely difficult in catfish aquaculture in those ponds because it's really tough to control the oligochaetes, I'm sure. Do you think over years, so we're probably, I'm assuming the same myxozoan communities are being maintained from one year to the next. Do you think that some of that could be driving some of these maybe not species differences, but genetic differences?
Definitely, possibly, for sure. I mean, a lot of these myxozoans, particularly Hennegaia delori, are endemic in the industry. And so there are some years where ponds break with disease and other years where ponds don't. And I think they're still trying to figure out why necessarily some years a pond will be fine and other, maybe it's just an amplification sort of thing, like each year sort of amplifying a little bit more till you get an outbreak. I think with the channel catfish, it's been it hasn't always been all in, all out. They've been grading channel catfish to always keep the industry supplied, whereas hybrid catfish aren't really good for grading. So now they're moving to hybrids that have the all in, all out. So that may also affect the myxozoan communities as well. So I do think some of the management strategies definitely impact the communities. I think some of the farmers are submitting pond water samples to screen for Hennegaia delori to see what ponds are really high in the parasite and circulating numbers of the parasites. So that way they'll avoid stocking those ponds. So there's definitely management strategies that have developed to prevent disease outbreaks. And that's why PGD is not as bad as it used to be in the industry, but it's still a problem. But to get back to your question, I do think that some of the management strategies do influence the myxozoan communities that we're seeing, for sure.
Awesome, thanks.
Oh, hi, Justin. This is Ben. Yeah, super cool talk. Yeah, I'm psyched that somebody's applying this amplicon sequencing to these systems. And I was curious if there's any other disease or environment that you'd like to apply this in, any other disease that might be caused by a mix, you think might be caused by a mixed community?
So I was trying to think about the different myxozoans that have substantial disease, like whirling disease and like C. shasta, and those seem to be pretty monomorphic, but you never know. There might be one or two parasites that aren't C. shasta that are causing an intestinal disease. So I mean, some of these breeds were lower in number, and I mean, Hennegaia delori and Hennegaia exilis were some of the more prominent ones that we saw. But I mean, I definitely think that there's definitely applicability for environmental screening for sure, to see what myxozoans are in the community, like in the environment, and see whether or not that reflects the fish hosts that are present. I think as far as disease processes, I mean, some of the bacterial ones for sure would be great to do like 16S sequencing, especially where you get sort of like the pylon, like we saw Flavobacterium talk earlier, just talking about the different strains that are associated, and they were doing challenge studies with Chrysobacterium. And so it would be good to potentially use metagenomics to see what the composition either of those cases is, or how it changes over time. So I definitely think there's a wide applicability for sure in other diseases, not even just myxozoans. So
yeah, oh cool, thanks man. Yeah, for species diversity or species discovery in an environmental system. That's super exciting.
Yeah, for sure.
Hey, Justin, this is Alex Primus. Very nice job, as always. Just kind of following up on that. I'm not that familiar with PGD and pathogenesis, but I wonder if there might be, some bacterial involvement, and whether it might be interesting to look at kind of the 16S profiles associated with those time points you sampled.
Yeah, no, for sure. I mean, we definitely see co-infections with columnaris all the time in these PGD cases. So yeah, no, for sure. Yeah, I think it would definitely be worth looking into as well.
Great.
All right, last call for a question. We've got just a minute left. All right, hearing none, Justin, you're done. Thank you very